Prapani sir, please go ahead.
Okay. So, good afternoon to you all. Thanks to Dr. Rangarao sir for initiating this webinar program. And also thanks to the other colleagues of Department of Biotechnology. So now I am interested on the phylogeny, and particularly I have been working with the gastropods. The same methodology I used to unfold the novel coronavirus evolutionary affinities. In this line, I am providing you the information, and here there are certain overlapping terms. One is phylogeny, the other one is taxonomy, the other one is systematics, the other one is the classification. So though all these four are overlapping, but there is a distinct demarcation among these four, with which we'll discuss at the end of the question hour. Now, the novel coronavirus that has emerged recently is not new to the biological system. Of course, the newness here is it is invading a new host. The new host is nothing but humans, and it is invading so rampantly that it spread across all nations, nearly 188 nations, and 75 lakh of people worldwide are infected, and about 3 lakhs of the people have lost their lives. And again, there is a recovery rate is also equally good. So that is why everyone around the world is interested, and the governments are also interested on this COVID activities. Now, let me go to the second slide. So I acknowledge sincerely the Google images, wherefrom I borrowed good many number of slides, and those are the things which I will be showing. Apart from that, my personal work on phylogeny I am highlighting. So the content of my presentation goes like this. The first one is the geographical eruption of novel coronavirus. The second one is types of coronaviruses. The third one is disease tracker. The fourth one is symptoms and risk factors. These are all the biological aspects of coronavirus. Unless we know these biological aspects, we cannot proceed the phylogeny, because the phylogeny is the one where we are going to link biological relationship with the novel coronavirus with others. Okay, the next one is phylogenetic analysis. The last one is the conclusion. With this, we'll be closing in another 20 minutes. Okay, the first one is geographical eruption of novel coronavirus 2. So as I was indicating to you earlier, the coronavirus is not new to the biological system. The newness appears only in attacking only the human host. So if you see this particular world map, majority of the viruses have taken their origin from East Asia and again the Middle Africa. So in this, if you take up just for the sake of the youngsters who are there as participants, chikungunya is one, Ebola is one, HIV one, Zika virus is one, yellow fever virus is one, and Japanese encephalitis is one, and dengue virus is one. Apart from this, the SARS. This SARS came into existence in the year 2002. MERS came into the existence in the year 2012. And then that is coronavirus, novel coronavirus has come into existence in 2019. And apart from this, we had Spanish flu somewhere in 2018. So, 1918. That is after a century later, we got very rampant viral disturbance in the society. So now this is the place where is Hubei province. In the Hubei province, that is almost Middle East of China, this has erupted. So this again, I collected this information primarily because Europe is the one highly disciplined country, and apart from that, they're all staying quite apart with the people. Even then they got infected. Here Spain, France, Germany, Italy, highly disciplined people, they're all away from the crowd. Even then they got infected, and now these countries are gradually receding, and now the UK, it is getting pronounced. So that is about the geographical eruption of this virus, just an introduction about coronavirus. The second one is the types of coronaviruses. Now, this is the structure of the coronavirus. Here, corona meaning these spikes all around. Okay, so here the spike is made up of a protein by a spike protein. Like that, there are 11 to 16 proteins have been reported in this coronavirus. Now, membrane protein, nuclear capsid protein, envelope protein, and this is a positive sense genomic RNA. It's all that we have been saying in the news regularly. And now these are a few coronaviruses that we have novel coronavirus. That's what I have been showing you in the previous slide. SARS virus and MERS virus. That is Middle East Respiratory Syndrome virus. Okay, for all these things, that some common thing that we're all observing here is the spike. Here is the spike. Here is the spike. Here is the spike. So the spike is a common signature in all these coronaviruses. That common signature has come from S2 domain of spike protein. Okay, this is what I want to let you know. Here, this is the spike protein sequences. This is the S2 domain. This S2 domain will help this particular coronavirus to get fused with the membrane of alveolar epithelial cells. Okay, so that's what it makes the virus to get endocytosed. And apart from that, here S1 subunit. This S1 subunit is having this receptor binding domain. This is the receptor binding domain with which we are getting infected. This is what this makes it to get anchored. This makes it to get anchored to this AC, that is angiotensin converting enzyme receptor, ACE2. Okay, so now let us see a few facts about all these four or five coronaviruses that we are dealing now. Okay, so this is Wuhan coronavirus, Middle East coronavirus, this SARS coronavirus, these are all alpha coronavirus, 229E, NL63, OCU, AHU, HKU, Hong Kong U1. Okay, now let us see these things. So this Wuhan coronavirus has come in 2019, Middle East has come in 2012, and SARS has come in 2002, and this alpha coronavirus is there all around the year, Okay, so now current status of Wuhan is it is spread across Wuhan, Asia, and also US and Europe. Now, all the continents now Wuhan virus is spread across. Now, if you take up the Middle East, it is there in Arabian Peninsula, and particularly 80% in Saudi Arabia. Now it is declining, it is slowly disappearing as the smallpox disappear. And here severe respiratory, the SARS is also gradually declining. No new cases are reported from 2004. When it came, it created a big havoc. So several people killed a lot of cats, civet cats also. Now, particularly this common alpha coronavirus, it is not dangerous, it is mild. Now, when we know that a few coronaviruses are mild, when you know that a few coronaviruses like Wuhan are wild, why not we use the mild one to cure the wild one, as our Edward Jenner and Louis Pasteur did? That's what our scientists must be doing, with which I am not familiar. This is just a projection to you all participants. Now, disease tracker. So here, this coronavirus, as it has been showing in regular TV and all that. So if you are infected on the initial day, we have to keep waiting till 7th day to get it confirmed that it is a coronavirus infection, COVID-19 infection. Or otherwise, we may get regularly fever, we may get malarial fever, we may get some other fevers, we will get normal influenza infection. So they are not get confused with this coronavirus. Okay. So on the 7th day, there will be shortness of breath. If anyone among us in the neighbors feel that there is shortness of breath, we have to suspect that it is because of the coronavirus infection. Not only this, there is also diarrheal problem also with this coronavirus. Not necessarily be the shortness of breath. If they are feeling anything related to the diarrhea, that also may be connected to this coronavirus, because I will let you know the reason why there is diarrhea, why there is shortness of breath next time. So here the infection is lung alveolar epithelial cells. Okay, they get infected. Once they get infected, what is happening is these cells are now totally totally laden with this, what is that called, the virus. Like this, it keeps spreading. See, once what is happening, as long as the virus is there outside the host, it is non-living. Once it comes to the host, it is living. So it is living means it is keep budding. So it buds so fast that it proliferates profusely, so that the viral load increases in the alveolar epithelial cells. Once it gets increased, then the host is victimized. So these are all various transmission electron microscope photographs. Okay. So as I was indicating to you, it is not only the respiratory distress syndrome that we are facing because of the NCOV2 infection. We are also facing the problem with the diarrheal problem. That's what luminal surface of intestinal epithelial cells are also getting infected, because these cells are also having angiotensin converting enzyme 2 receptors like that of the lung epithelial cells. Okay, all that blue thing which are being showing here, same thing. They're all because of this type of the infection that is there with the luminal surface of intestinal epithelial cells. Okay, now the symptoms and the risk factors. So the systemic symptoms all over the body: fever, fatigueness. So the next one is the respiratory symptom is the dry cough, and shortness of breath. Particularly, whenever we infected with influenza virus, chikungunya virus, and other, we never feel the shortness of breath. Here, specifically, that's what the doctors say. Since the beginning, governments are also planning to have more number of ventilators. That's what our Vice Chancellor sir is also telling. The automobile companies are shifting their manufacturing units towards this ventilator manufacturing. That's what the shortness of breath. And circulatory system is also, there is a problem. Again, the kidneys, as I was mentioning to you, lungs are being affected, intestine is being affected. Not only that, kidney is also being affected, because kidney is the site where from the angiotensin is being developed. So there is angiotensin, renin, vasopressin. These are all the hormones that are being regulated, and they are being produced from the kidney. That's what kidney is being affected, because at the later stages of COVID-19 infection. Now, so this is what the observation made in China, in Hubei, that people are also having this diarrhea symptom. 48.5% of the people suffered with the diarrhea. They must not have shown the, what is it called, respiratory distress syndrome. Now, so this is a sort of a recent, what are the precautions that we have to take? Recent travel to red zones we have to avoid, and other places where there is an outbreak. So these are all the guidelines given by every government, and locally also it has been imposed by several police and other people. The next one is close contact the people who are diagnosed with the disease. Unfortunately, our doctors and paramedical staff are coming very close contact. They are getting victimized. Close contact with anyone who has visited an outbreak zone, and contact with secretions or fecal matters from an infected person. And again, eating or handling wild animals. So these are the things we have to avoid. So this is the crowd. So whenever we are in the crowd, there's every possibility for that. That's what in the beginning, India has not faced this problem like that of the China and other countries, because our metro traffic is less. In other countries, metro traffic is heavy. They're all got infected there. And now with this background information, now we are coming to the phylogenies. So as I was mentioning to you, there is systematics, there is taxonomy, there is classification, there is phylogeny. These four, though there is overlapping, but phylogeny is altogether different from all these things, because in nowhere in the taxonomy systematics, interrelationships among the taxa is being shown. In phylogeny only, we can build the interrelationships among the taxa. Among the taxon. Okay, so that's not only the interrelationships, even evolutionary relationships also we can speak. Apart from that, the recency of their origin also we can speak in this phylogeny. Okay, as a result, the phylogeny provides a phylogenetic tree. The phylogenetic tree is a model, is a diagram that delineates the evolutionary affinities. Those are the things we are going to discuss. Now let me tell you what are the works that I have done in relation to this phylogeny of novel coronavirus. So phylogenetics in biology is the study of evolutionary history and relationships among individuals, traits, and groups of organisms. See, I'm telling you about the traits, not only the taxa, even the traits also we can work out. That is metallic proteins, evolution of metallic proteins we can speak here. Evolution of hemoglobin we can speak here. Evolution of hemocyanin we can speak here. So like this, traits also can be discussed through this molecular phylogeny. And even the population also we can discuss. That's what in the next slide I will be showing about the population also. Phylogenetic inference methods are used to evaluate observed heritable traits, and the results ultimately lead to the hypothesis. Okay, now let us say the typical phylogenetic tree looks like this. Typical tree wherein A taxon, B taxon, C taxon, D taxon, E taxon. All these taxons are not there in one cluster. A and B is one cluster, so they are sister related taxa. C and D is one cluster, they are sister related taxa. So this C and D have become non-sisters to AB, non-sister related taxa. Okay, so AB is one primary cluster, CD is one primary cluster. These two clusters are again grouped as a tertiary cluster. So this tertiary cluster are sharing the common features. As a result, it constitutes a clade. So the total clade has taken the origin. That's why it's a rooted tree. It is a rooted tree. And apart from that, there is an outgroup. From the outgroup, it took an origin. So that's what here, if you see this branch length from this to this, it is from ancestors to present day we can trace. But this is a dendrogram, it is not a phylogram. In the phylogram, the branch lengths are varying. In the dendrogram, all lengths look alike. Okay. So I wish to let you know the importance of this phylogenetic tree. If you go to any of the journals which are there handling the biochemistry or molecular phylogeny or bioinformatics or genetic engineering or immunology or any virology, nearly 40 to 50% of the publications and every journal will be having this phylogenetic tree displayed. That's what the importance of this phylogenetic tree nowadays. Okay, as a result, in our Vignan University, we are having a course for 48 hours for BTech students on molecular phylogeny and evolution. We have given much emphasis for this. And apart from that, you please have a look at this. This is borrowed from GCID, and these people have done a lot of work. That is, they collected from all over the world, the genome sequences of our coronavirus, and they compiled and they made a tree. So here you see, this is not between the taxa, it is between the members of coronavirus, between the members of the coronavirus from India, from China, from several places. So as a result, they found out nearly 36,000 genome sequences. 36,000 genome sequences so far have been isolated from coronavirus. None of the other species have done so far like this. And apart from this, out of these 36,000 sequences, they identified S clade, L clade, V clade, G clade, GR clade, like this, all of them each. So that's what I wish to let you know here is, it's not only between the taxa, between the individuals, between the traits also we can work this type of the phylogenetic tree. Now, so for building the phylogenetic tree, so the morphological characters will not help now. So those were the days we were using the morphological characters to build the phylogenetic tree, a dendrogram, using numerical phonetics. Now numerical phonetics has already been outdated, and that can be given as an exercise for the students to learn what is the phylogenetic tree. Now, if at all you want to build a phylogenetic tree, we require a sequence. The sequence we can get from like NCBI, like the DDBJ, and all that we can procure the genome sequences. Okay, so the genome sequences are several formats. So it may be FASTA format, it may be Nexus format, it will be mega format, it will be Phylip format. This particular one that I have been showing here is the mega format. Okay, so it is the protein sequence. The other one is also protein. For those who are not familiar, new students, I am showing here. This is also protein sequence. This is the DNA sequence in mega format. Okay, so like this, we have to select the the sequences of our choice on which we are working. So the phylogeny, whenever you start working on the phylogeny, this is the crux. This is the point of origin of your work, from where you can develop the hypothesis. Here, if you make anything wrong, ultimately our tree also will be wrong. Okay. So for this to work, we have got this many softwares. Not only this, several softwares are there. I have been working with mega software. There is Ruxmal also our students have done, Mr. Base also our students have done, Phylip, Cluster Omega, POP, Persuages is a commercialized software, all others are free, and Tree Finder. So in this, Mega is the software which was developed by one of the Indians by name S. Kumar in collaboration with Japan and USA people. So now let's come to the point of my research work. I can say my research work, the hypothesis. Here, null hypothesis and alternative hypothesis, two hypotheses we have to project whenever we are doing in the research. In this, novel coronavirus is not related to the coronavirus, that is bat coronavirus, and not found to be recency in its origin. Okay, that is the null hypothesis. The alternative hypothesis, novel coronavirus is related to bat coronavirus and found to be recent in its origin. So we have to select any one of these things after obtaining our results. Okay, now let's come to this point. So this is the genome of coronavirus. This is the genome of coronavirus. Oh, So this is the genome of the coronavirus. This is the genome of the coronavirus that I have taken. Okay, so the genome of the coronavirus is approximately 30,000 nucleotides. That is, it varies between 27 to 30,000. So it is among the species that I have chosen. Let us see. I have shown you the mega format that I have chosen, FASTA format that I have chosen, and the software that I have taken called the mega software. And now, after having submitted all these things into the mega software and maximum likelihood method, then I have procured this type of the graph. This is not the graph drawn with the pencil on my own. It is not the graph made on the paint. It's not the graph made on any type of the computer system. It is the graph model. This is the model given by the FASTA format for us to interpret it. So here, once we go to this, it is the bad SARS has come as a root. Okay, so this is getting related to the SARS coronavirus direct. Both of them constituted a cluster. That's what in the beginning I shown you, A taxon, B taxon, like that. Okay, so from this it is getting connected to this Wuhan CoV. That means this pair of cluster, from this pair of cluster, there is an offshoot. There is an offshoot. In that offshoot, there is Wuhan. That means Wuhan coronavirus is related to either SARS coronavirus and bad coronavirus. To which extent it is related, next slide we can see. Next we will see. So here the MERS coronavirus is also closely associated with the next offshoot. Not easy. This offshoot and this one both have not formed a primary cluster. Clear? The other one is alpha coronavirus. These two are the alpha coronavirus. That is 229E coronavirus and NL63 coronavirus. These two are alpha coronavirus. Both have constituted a single cluster. That means it is away from the tertiary cluster of beta coronaviruses. Clear? Now, in this also you make a note. In this also please make a note that this is not a dendrogram. It is a phylogram. In this, the length of the branch of all these things, make a note. Here, this branch that has appeared here is a short branch. The short branch means the recent origin of this coronavirus is reflected through this phylogram. Now, this is another phylogram has been made, not using the genome, but using the protein sequence. The protein sequence here is the spike protein I have taken. Spike protein I have taken. And the spike protein sequence is like this. And it is having both S1 subunit, S2 subunit. Taking the spike protein into consideration, this type of the graph has come. So these two graphs are ditto. There's not any risk. So the protein, you're all familiar what the protein is. Protein contains amino acid. And again, it is having more than 11 types of the proteins, of which one protein we have taken, this is called the spike proteins, which is having around 1100, what is that called, amino acids. But if you take the genome sequence is nearly 30,000 nucleotides while comparing all the 6. And this 1100 amino acids while comparing all the 6, we got the same type of, what is it called, phylogram. That's what I'm going to explain now to you. So in this, again, if we take up the BAT spike protein and the SARS coronavirus spike protein, both constituted one, what is it called, the cluster, primary cluster. Okay, from this, there is another offshoot, that is the novel coronavirus offshoot. Okay, then from this again, one more offshoot is called the MERS. That means these 4 belong to the category of beta coronavirus. They all constituted one tertiary cluster. This is one primary cluster. These 2 are the offshoots. They constituted one tertiary cluster. Okay. The next one is the alpha coronaviruses. These are the 2 alpha coronaviruses constituted one primary cluster. In this, I wish you all to focus on the branch length. I wish you all to focus on the branch length. See this length, see this length, see this length. So while seeing all this length, this novel coronavirus length of the branch length is short. This short branch length indicates, okay, it is recent in its origin and its relationship with other, what is it called, the coronavirus will be shown here. Okay, so with this we'll go admit. Okay, so the next slide. So here the number of amino acids, as I was mentioning to you in the spike, is approximately around 1100s to 1200s, and S2 length is around 600s or so. And S1 region is the one which is having the receptor binding protein. It is also around 200s or so. And here, as you say, BAT is also like that of the novel coronavirus. It anchors to ACE2, angiotensin converting enzyme 2. Only thing is MERS is different. It is DPP. that is dipeptidase peptide. The dipeptide peptidase, it is binding to that site. Okay, now the percent identity matrix. So here you please make a note that this is what our novel coronavirus. This novel coronavirus is related close to this bad coronavirus, and again this SARS coronavirus. That's what is 76, 77. But when you consider this alpha coronavirus, it is 27%, 27% related. Even with the percent identity matrix that we build up through Omega Cluster Omega software, online software, we have developed the close association of novel coronavirus with bat and SARS. Go to the next one. So apart from these things, we also did use this IEDB, that is Immune Epitope Database, IEDB, Immune Epitope Database. In that, we submitted the sequences, protein sequences, and we wanted to develop the epitopes for this. Okay, so in that epitopes, we got nearly 64,000 hits. So 64,000 hits later, we have taken this Excel sheet. There we have filtered that. After having filtered, we have taken 10 of these epitope sequences. These 10 of epitope sequences, we submitted this conservancy analysis. In this conservancy analysis, the bad COV and NCOV and SARS COV, these are all sharing the conservancy. The other things, if you take MERS and 2T and NL63, they are less than 50%. So all these epitopes that we have selected here, 10, they can constitute a multi-epitope vaccine. So this multi-epitope vaccine, if you make, it can suit well for this coronavirus infection, and also it can suit well for future, if at all there is any SARS again reappear, it can also be worked for this. Okay, so this is a cluster analysis also we have done. All these epitopes are interrelated. They share their identity. That's what a line comes between two of these peptides means. It appears that all these peptides share their identity. As a result, they can be used for developing the multi-epitope vaccine. So the conclusion. So the DNA and protein sequence of novel coronavirus reflected that it is showing molecular affinity with bad COV and other beta coronaviruses, as they all constituted a single tertiary cluster. Now people are coming viral, what is it called? YouTube also has come that this corona is not virus, it is a bacteria. Okay, so now with the proof we are showing that it is not the bacteria, it is the virus. And they are telling in the YouTube that it is bacteria, it is causing blood clot. Blood clot can be cured with the aspirin. So aspirin is not related to work here to conclude, and blood clot is not related to conclude. It is only the coronavirus with receptor binding domain binding to the ACE2. That is what the crux that has been identified, designated here. The other thing is, apart from that, it constituted a single tertiary cluster with the beta coronavirus. That is most important. So it is not sporadically gets spread across the coronavirus. Alpha coronavirus is separated, and beta coronavirus, it is not physically we separated them. Only the nucleotide sequence separated them through this dendrogram, through this phylogram. So that is the importance of this type of the softwares, that we can assess the evolution relationship and molecular affinities among these individuals. Apart from that, the branch length, what I have shown you through this diagram, also is sharp, that appears that it is coronavirus is also recent, it is telling. So that's what we have been telling. Coronavirus is not new to the biological system. The newness is only the infecting human beings. Okay. As a result, the null hypothesis is rejected and alternative hypothesis accepted. Okay, apart from that, this is about the DNA matrix. Here, there is a very possibility that we can have the multi-epitope vaccine with that type of the peptides, so that we can get cured this NCO, that is novel coronavirus, so that from next year onwards, no one suffers from novel coronavirus. Okay. So the immune boosting is the only solution hereafter to get ourselves protected from this type of the viruses. And a good many number of papers are there. Rangarao sir has advised me to put a few papers here. Molecular evolution analysis people are doing from various, that is from protozoa, from bacteria, from viruses, from actinopterygiae. Several of those things people are doing, and even on primates also people are doing, even Aryan population and Dravidian population, taking the genome into consideration. And like this, there is again the evolutionary perspectives based on genome analysis, which is that work done by several people outside. And we also have done some work in this COVID break. So thank you to you all. Any Rangarao sir, I'm waiting for the questions, sir.
Yes, sir. Thank you, Professor Prapani sir, for giving a wonderful lecture on this COVID-19 enlights about coronavirus types of disease trackers and symptoms, risk factors, phylogenesis analysis, evolutionary history, spike proteins in COVID virus, and genomics in COVID virus. Thank you very much, sir. Now, actually, open for the discussion. Maybe a few questions can be asked participants.
May I ask a question?
Yes. Welcome, John Kenneman, sir. Sairam.
Thank you. I'm curious. The result that you found is that it's recent and that and SARS-CoV, and that the jump to humans was very sudden and, as we can see, very effective. I would think that it's important to find the intermediary species, but I don't see that anyone has found that yet. How important is it to find that intermediate species? And I'm wondering how it could evolve so well to infect humans. And it would seem we would need to identify it in the intermediate species in order to know that.
Okay. Here, thank you, Dr. John Kenneman, sir, Professor, sir. So, with regard to the intermediate species, that term doesn't pertain here, sir. Here it is a mild species you can take. There are mild species of coronavirus and wild species of coronavirus are there. Okay, mild species are these alpha coronaviruses, and this wild species are the beta coronaviruses that we are coming across. Intermediate means in biology we speak that intermediate host, that is another in between, another specimen make it get infected like that. There is no question of intermediary involved here. There are mild and wild, both are there. Let our scientists have to work whether using this mild, whether this wild can get cured by developing any vaccine. Still it is under the trial, sir.
Okay.
And another important aspect here is the mild one is not anchoring to ACE2 receptor, angiotensin converting enzyme receptor. But with regard to the wild one, it is getting anchored to angiotensin converting enzyme receptors. So with regard to this virus getting entry into the human beings, the paths are different. Mild one path is different and wild one path is different. Of course, if these two, even though these two are different, there is a very possibility for making a vaccine from the mild one to cure the wild infection.
Nice. From what you have seen, it appears to be a natural evolution.
A natural, it is natural evolution. It is natural, sporadic, random.
Yeah. Thank you.
Thank you. Thank you to see you on webinar.
Okay, next, anyone questions? Any questions from the participants? Okay, now let's move on. Second speaker.
Thank you, sir.
Sir, can you make me host? Professor Krupanaji, sir, can you make me host?
Stop sharing. Host.
Sir, go to participants.
Yeah, go to participants. Yeah.
Sir, go to participants.
Yes, sir. Then.
And then, and your name is there.
Rangarav. Just to present Rangarav sir name, sir, go to more.
More, go to more. Very good, sir.
Yeah, make a host. Make a host himself.
Fantastic, sir. Well, we learned through you, sir.
Yeah. Right, sir. Right. Thank you.
So
Rangarav sir, you can proceed further.
Yeah, thank you very much, sir. Thank you.
Yeah. Thank you, Krupanaji, sir.
Thank you.
Okay, now let's move on. Second speaker, Dr. Obul Reddy from Germany. Sir, Obul Reddy, sir.
Yes, Rangarav, I'm ready.
Yes. So, so now actually, Dr. Obul Reddy actually going to talk about the role of genomics in COVID-19. Dr. Reddy actually received PhD from Humboldt University, Berlin, and Dr. Obul Reddy has various experience in research, and also he had a postdoctoral experience and several positions actually hold previously. And Dr. Reddy actually expertise in translational research, cancer genetics, next generation sequencing technologies, and functional genomics to identify cancer predisposition, genes, and development of risk stratifications markers. Actually, Dr. Reddy published more than 50 research publications and has several national and international conferences attended, and he has research grants, and he actually awarded several fellowships. Now, we request Dr. Obul Reddy, sir, please give a talk on the role of genomics in COVID-19. Please welcome you, sir.
Thank you very much, Dr. Rangarao, for the kind introduction.
I will make you host, sir. Please wait.
Yeah.
I will make you host.
Did I get? No?
Yes, sir.
Okay.
Please.
Okay. Okay, good afternoon, everybody. First of all, I would like to thank Professor Kaukishore Garu for kindly giving me the opportunity to share my experience here, and also thanks to Dr. Jalija and Dr. Rangarao for being in touch and making this possible. And before going further, I would like to say that I'm not an expert in virology. I'm a translational molecular biologist who's having experience in translational genomics and functional genomics, and I just thought of sharing my experience how genomics can be applied to COVID or any kind of viral diseases. This can be applied to that topic. So briefly, as Professor Krupanidhi sir has set ground for me, I don't have to go in detail for the background of the viruses, so I will run through briefly. So coronaviruses are a large family of viruses that are known to cause illness ranging from mild cold to severe diseases such as MERS, like Middle East Respiratory Syndrome, and severe acute respiratory syndrome, SARS. So those were the main acute ones. The other ones are mild, so that we may not even notice that we have some kind of virus. And the novel coronavirus is a new strain of the coronavirus that has not been previously identified in humans. And the Chinese scientists sequenced from the earliest patients in Wuhan, that was at the end of 2019, and they have compared with the coronaviruses and identified it as a new human virus and named it as SARS-CoV-2. So brief structure of coronaviruses. Coronavirus or any viral structure is a group of enveloped positive stranded RNA viruses there, and they consist of 4 structural proteins, including one of them is the spike glycoprotein, and envelope protein, and a membrane protein, and nucleocapsid one. Among these 4, spike glycoprotein is the most important surface protein of the viruses. That is the one which is interacting with the angiotensin converting enzyme 2, which Professor Krupanidhi was introducing. So that's the important one. Coming to the types of coronaviruses. So there are how many types of coronaviruses? There are 6 strains of coronaviruses that have been identified so far. So the first 4 are not so pathogenic. They cause mild respiratory diseases. SARS-CoV and MERS were the 2 large-scale pandemics that have been gone through, and now recently, not even before that, we have found proper treatment for SARS-CoV and MERS. We came up with this SARS novel coronaviruses, which is more faster than the other 2 viruses. Briefly about the state of the art about COVID-19 in India, using an integrated genome view of SARS-CoV-2, Pramod Kumar et al. From National Center for Disease Control and CSIR Institute of Genomics and Integrative Biology, they have sequenced 104 patients from throughout India, from different states, and they have found that, we all know that it has arrived in India during at the third week of January or something like that, from coming to Kerala from Wuhan, China. And then March 2020 onwards, different cases arrived from rest of the world, like coming from Italy and from Indonesia, and triggered further outbreaks in India. So in this analysis, they have analyzed, as I told, 104 patients from different parts of India, and they tried to understand the introduction and how the local transmission worked. From their analysis, they have shown that there are multiple introductions, as we know from the news also. They have shown it scientifically, and they show that it has arrived from Europe and Asia following local transmissions. So they saw 3 waves of transmission, the first one coming from Wuhan, and then the other ones coming from other parts of the world, coming from Indonesia and Italy and other parts of the world. So these are the results. Probably this is under the results are freshly in bioRxiv. They are still working on it to publish somewhere. So to look at the structure of COVID-19 and other viruses, here is the phylogenetic structure of the viruses. Here you can see novel coronaviruses is closely resembles with SARS and MERS, and these are the 3 severely affected diseases, and they are very closely associated. This is just to show that how they are phylogenetically related. And the structure of the COVID-19, all of them 5' to 3' UTRs, all the 3 variants, they are all having the same 5' UTR, and also the gene part, and only you can see some changes in novel coronaviruses highlighted in red here in the 3 spike protein and 3' part. These are the main differences. It's very important to know how the viruses are differing from one to another in identifying kind of vaccines or if you want to find therapeutic regimens. So what is the view of genomics for COVID-19? For example, whole genome sequencing provides the highest possible resolution information about an organism's genome, and this can be used for designing, finding the epitopes to which you can target the vaccines and develop any therapeutic approaches. And this will also help in infection transfection management, how it is prevalent, how it is transmitting from one place to the other, how it differs from one place to the other. And also by analyzing differences in the genetic code of the viruses from different patients, it's possible to map the spread of the disease in real time, like from where to where it is spreading, how it is spreading with the migration and people movement, and you can track new mutations. If it is changing with the mutations, the severity of the disease is changing or it's getting milder, you can predict in real time and can develop treatment approaches in real time fast. And better understanding of the genetic makeup of the viruses ultimately saves lives, because you can quickly identify what are the pattern of spreading, what are the mutations, if the mutations are leading to severity or mild conditions, so that you can reassess and reorganize your priorities and approaches, where to put your efforts and where you can take some things easy like that. So how the whole genome sequencing can be carried out or will be carried out? You can there is a new initiative in UK where they have there are no proper facilities, especially set up for this purpose only. So they are using the existing genome sequencing facilities. They collect samples from different parts of the country and send to different laboratories, so that you can use all the available resources and get the things done in quick time. And then you can use all the methods available for whole genome sequencing. And then they come to the conclusion that long read sequencing, like using the nanopore technologies, will be better, because it gives long sequencing ranges, and it can also be used that has been also used in other outbreaks like Zika virus and Ebola viruses. So how does it help doing whole genome sequencing? By sequencing the whole viral genome, researchers can pinpoint the genetic changes that occur in the viruses as it spreads through the population. And this approach is useful to understand the transmission of the viruses, how the genetic sequence of the viral genomes change from place to place, and also how they build the family tree, how they're closely related, like Professor Kripanidhi told, now how they're closely related to MERS and SARS, and also the other researchers have shown this is possible quickly to do with whole genome sequencing. And also you can monitor disease spread within and between populations over time, how it is spreading. And also it helps with identification of infection hotspots. If it is specific to a place, you can identify if there are any certain mutations in this area that are causing the virus to spread more prevalent than in other places, like super spreaders and so forth. And you can also design treatments and vaccines based on the sequences, because understanding the viral DNA sequences will assist researchers in designing therapies and vaccines that target specific features of the virus, because one fit for all will not work in this kind of viruses. So you have to design viruses, vaccines and therapeutic regimens that can specifically target these viruses. So knowing the epitopes and the sequences or mutations is very important in designing this kind of approaches. And also you can monitor the viral evolution, the genetic changes that are fast changing, and virus is also changing, fast changing. It's a genetic map, so you can see them, they are becoming more virulent or sometimes becoming like mild. So you can find the early warning of virus emergence or treatment resistance with the help of whole genome sequencing, so that you can prioritize how to deal with the situation and prepare for the future. So the infrastructure and protocols that are created through this project, this is like nobody has we're ready for this kind of situation. Now we are all slowly trying to learn and trying to adapt to the new systems, and people are finding different people and the combined effort finding ways to tackle the situation. So the effort and the protocols and the disaster management system, you may call, will help in getting ready for further pathogen outbreaks in the future. We can maybe able to tackle them in a different way than what we are doing it now. But what are the challenges and outstanding questions? There's a lot of potential to make huge advances in understanding of the viruses by knowing the genomic sequence and differences and mutations and the comorbid conditions and so on and so forth. But it wouldn't be easy, because whole genome sequencing is a powerful tool for understanding the viral transmission, but DNA sequences alone are not enough to track viral spread without another information, like how contact tracing, like with how many people this person has been in contact, so that you can contain them and control the spread of the disease, and you can also follow up with them. So it is as a combined effort of all the other measures that are available, but whole genome sequencing will give first step to identify the sequence, like evolution and the mutations and so on and so forth. And for example, best example in Germany, in February there was a virus case in Munich. This person got in contact with a scientist coming from Wuhan, China. So they didn't know. They thought, then they found out this person and they contained, and they thought that okay, they controlled it. Then there was also a couple who visited Italy and they came back to Germany and they participated in a carnival. So they thought it's also like Munich and the same origin. They didn't anticipate that it will go to such an extent. Then with this, it spread so rapidly. Germany was coming to like almost top 5 situation. Now it has come down, but it was kind of a panicky situation. So this implied that the viruses at the source of the Italian outbreak may have originated in Munich, though it was also considered equally likely both the viruses were originated in China, but they thought that they separately arrived in country. But we have seen that in Italy also, and also in France and Spain, it's like behaving in different countries in different ways. So knowing the genomic sequence and how it is changing would be important, and it can tell us more about the changes and how one can tackle those situations. So one example is COG, COVID Genome UK Consortium, is an example of the use of genome sequencing technologies. While it is exciting, genome sequencing of the virus for COVID-19 is only one tool, because we have to do other technologies like rapid testing to confirm suspected cases, as well as to determine if their previously infected individuals have immunity. Like if their immune system is good, they are more crucial. They will not show. On one hand, they may be healthy and asymptomatic, but on the other hand, they will be the carriers, like they're infecting many people without knowing that they have the disease. So that's very important to find out the other symptoms and also approaches in addition to the whole genome sequencing. So in context of this UK consortium, this consortium may face several technical challenges in terms of analyzing viral DNA. For example, some sample types such as nasal swabs don't always provide good quality RNA. That may be giving good quality and it is not giving good quality. So it's like trial and error. They are doing a lot of, but nevertheless, it will be a potentially vital role to play in mitigating the long-term impacts of the current outbreak, and further it helps in improving our ability to understand, manage future outbreaks if there are any. Hopefully not, but if there is something, probably this will help in finding ways to tackle easily. So what's the role of individual risk of for COVID-19? Every person has a vital role to play in reducing the spread of the infection, as we all learned from the experiences. Should a person become infected, the individual risk of disease is highly variable. It depends on the people to different extents, because some people are highly likely or some people are less likely to become infected in the first place. This may be because of their response to infection or their immune situation, and also several other factors, comorbidities, etc. Usually, most risk are the youngest, oldest, and those with pre-existing diseases. But these patterns can vary. The swine flu pandemic, for example, in 2009 proved unexpectedly dangerous for otherwise healthy pregnant women, while the notorious 19 Spanish flu was unusually devastating because it caused severe disease and death in young, healthy adults. So it's very important to know how the genomic structure and also the other conditions that play a role in disease onset and their severity. Why some groups are at higher risk? People classified as high risk should be particularly stringent in efforts to avoid SARS-CoV infection, are like those above 60, or basically above 60 and 70 and older, and pregnant women, and very obese, and those with significant medical conditions such as diabetes, neurological disorders, heart, lung, liver, and kidney diseases. They are less likely to resist infections because the immune system is maybe not so effective, and also it maybe typically decreases with age, and also during pregnancy the immune system is weak, and also to cope up with the physiological effects of infection. And obesity can both weaken the immune system and also worsen the respiratory stress. That's why obese people should also be very careful with this situation. That's how it could explain why patients who develop severe diseases are more likely to be overweight or obese. That's another thing. And the unexpected risk group, people from high risk groups account for the majority of deaths, but the younger and apparently healthy people who develop severe disease, and sometimes they may also die. Why do these low risk individuals suffer so badly with a viral infection that in others causes mild disease or even no appearance of disease at all? This is the very important group, and it's worrying group because they don't know that they have an infection and they don't show any symptoms. They'll be going out and meeting many people. That would be like a moving bomb. So it would be very important to know the mechanism behind these people, and also environmental factors like healthcare professionals who are exposed to the virus for long periods of time, and also those people who are working for a long time with the stress, they become very ill and die. Yeah, those are the conditions. They cannot fight infection because of a high load of viruses. However, why a healthy person should develop severe disease and die in this instance? It's very likely that human genomics play a significant role. If we can identify why they get severe viral infection without showing any symptoms, and why they will die. This will help in identifying and stopping the spread of the disease majorly. And any changes in the viral genome that affect risk, this was almost certainly a genetic change that is enabled the virus to make the entry from infecting animals to the human transmission, the case of now in COVID-2 also. So that's very important to find what makes the transmission from animals to the humans. However, viral genome analysis has so far suggested relatively very little variability between different strains of SARS-CoV-2. So maybe we will find there are many groups, many people throughout the world working. Maybe they will come up with some approaches which are common or which are different, and we may find a lot of things in the near future. So I hope we will be ready to tackle the diseases in future further. Human and also the human genomic variation, it is different. Like for example, if you take Asian population and Western population and European population and non-Finnish European population, there are different, like the SNPs are also different, and also they may affect the severity of the disease. So with the sequencing efforts coming to an end, probably we will all the major genome sequencing efforts around the group will shed some light how all this background information play a role in the disease severity and treatment options. Until such collaborative research efforts are advanced, we can't know sure for what genomic features may contribute to the disease, but I'm sure that we will learn a lot and great deal of them in the near future. So we have to wait and see. So to set the unexpected risk people with genomic variants that affect their immune system, responses are more susceptible. That's one thing known. And individuals more susceptible to respiratory symptoms, which are associated with severe disease and death. And also resistance and susceptibility to viral infections is often associated with surface proteins through which the virus gain entry into human cells. So these are the main things, and the main possibility that has been proposed, and also Professor Krupanidhi had said that angiotensin receptor enzyme 2 is an important gene, and any variations in or mutations in this gene may also play a role. So we will learn this. In addition to that, how artificial intelligence together with genomic tools help to combat COVID-19. There is just the NVIDIA has released a new artificial intelligence model and also a genomic sequencing software and speech recognition technologies for the medical community that can help in tackling the COVID-19. And they have also made a computational genomic software called Clara Parabricks that can give the real-time whole genome sequence under 20 minutes, which would be very important at this time. And they have also introduced a GPU-accelerated RNA sequencing pipeline that returns results in less than 2 hours, and providing researchers with critical insights about patient susceptibility to disease and disease progression and response to treatment. This will be really important in finding therapy strategies and things. With this, I would like to say thanks for the organizers for giving me an opportunity to speak. And I also would like to say that all this knowledge is not anything related to my intellectual data. It's all collected from Google and publications just for the educational purposes of the students. I'm really happy to take if you have any questions now. Thank you.
Dr. Ogunreddy, sir.
Yep.
Thank you very much for giving a wonderful presentation, sir, and enlightening about phylogenetic tree of coronavirus, structure of COVID-19, other viruses, understanding the transmission of virus, and design treatments and vaccines, how we are going to move on, and monitoring viral evolution, and then genomic is one of the weapon is to fight this COVID-19 and challenges and then outbreaks, and also artificial intelligence genomic tools to help combat COVID-19. Sir, thank you very much, sir, for giving you wonderful information. So now open for the discussion. Participants, if you have any questions, now you can ask. Yes. Vinod Raj. Hello? Hello? Dr. Vinod Raj. Hello, can you hear me? Sir, can you unmute, sir? Verbal ready, sir.
Yeah. I'm trying to see to whom.
Dr. Grace Vinod Raj. And Dr. G. Durgaya. They have actually a few questions, sir.
Yeah, I'm trying to get that.
Yeah. Can you please ask Dr. Vinod Raj, sir? Yes, you can post, please. You can post, sir. Dr. Vinod Raj, sir, you can post. Ready, sir. Yeah, actually, Dr. Vinod Raj, actually it is giving a background noise, sir. Hello. Hello, I request all the participants mute your sound. Sir, Dr. Obuledi, sir, unmute all this one.
I'm doing that, but I'm doing it.
Then you can click escape and then you'll quit that particular presentation thing. You can click escape and or probably press Alt plus F4.
I request all the participants unmute, mute all your sounds. Okay, Obuledi, sir, can you host me? Can you please host me? Sir, go to participants.
Yeah.
Rangarao, Dr.
Yes,
Rangarao.
yes, sir. Thank you.
I could not unmute anybody except I could change. Now, please, maybe you can give the access.
Yes, sir.
I have a question, Dr. Obuledi.
Sir, Obuledi, sir.
Yes.
1 minute, sir. Professor Kavkishore, sir, have some questions. Yes, sir has some questions. Wait, sir.
Sir, I couldn't hear anything.
Can you hear me now?
Yes, sir. Very feeble, but yeah, I could.
Okay. Do they leave any toxins in the body? Do these viruses leave any toxins in the body? Once the infection is relieved, these toxins are likely to continue in the body, or what will happen to them?
Sir, could you please repeat? It's hardly audible. I could hardly hear you.
Okay. Can you hear me now?
Yes, yes, sir.
Yeah. Do these viruses leave any toxins in the body, human body?
I really don't know, sir, but probably there may be. I don't know this. I'm sorry, but
okay.
Yeah,
fine.
Not in my knowledge.
Sir, Dr. Obuledi, sir. So one of the participants actually asking question.
Yeah.
That as most of the German population has pollen allergy, does it play a role in infection of SARS-CoV-2?
Very good question, sir. Yeah.
Do you think will it be dangerous for allergic patients?
It's a very good question. Very good question. I think almost half of the population in Germany, at least, are allergic to pollen. Unfortunately, I'm also one of the affected after coming here. it doesn't look like that. If you see the German population, at times it looked like people are getting infection very fast, and they thought that it's going to affect. The predictions were like 60 to 70% of the population. Yeah, maybe we don't know exactly. Not everybody is tested like in other countries also, but the social distancing norms has given very good results in Germany, and the cases have really gone down, and they have effectively controlled as of now, and they are now started releasing people to go out, and then they are warning that we may have to be ready for the second wave of attack. But as of now, there is no known role of pollen allergy. However, DKFZ is conducting a pilot study with a participant from our institute, and they are trying to monitor the situation for the 6 months to see all these kind of things. What are the effects, and how people are, healthy people are coming up with the virus positive. They are doing all kind of tests. Maybe in next 3 to 6 months, we'll get more results on this.
Thank you, sir. Sir, another participant, so we have a question. Why
Yes.
those people will affect badly with COVID-19?
Sir, how do?
Why
Yeah.
most people will affect badly with COVID-19?
Why old people?
Yes, yes, yes.
Yeah, as I told, maybe one thing would be their immune system is weak, sir. With the increasing age, the immune system is becoming weak, and they are more prone to infections. The body is not able to fight the infections. That may be the reason. And they may also have comorbid conditions. They have either BP or like lung or kidney or heart or diabetes and overweight. Maybe any of these other comorbid conditions may be playing a role.
Okay, thank you very much. So there is one more question.
Yes, sir.
That after getting virus, will this show any effect on their next generation?
Yes, this is also a question that maybe this is one step ahead. Whoever is asking, very intelligent. There were people saying that whether the viral infection, people get infection and then they get healthy and they may have long-term impacts. This is the immediate question. But the question now, whoever is asked, we went one step ahead. It's very interesting. We don't know it, sir. Maybe we have to wait and see.
Okay, thank you very much, sir, for your this nice presentation, and thank you for accepting our invitation. Now we will move on another speaker, sir.
Thank you, Dr. Rangarao.
So next speaker, we are welcome Professor Rahul Narang, Department of Microbiology, Mahatma Gandhi Institute of Medical Sciences, Maharashtra.
Hello, good afternoon.
Sir, Dr. Rahul sir, can you hear me?
I can hear you very well, sir, but then I'm not able to share my screen.
No, sir, I will host you, then you can able to share the screen.
Okay,
sir.
All right, sir.
Yes, sorry for that. Yes, sir. I host you, sir.
Yes, sir.
Please go ahead, sir.
Yes, sir. I think my screen is visible, sir.
Yes, sir. It is visible.
Okay, sir. So thank you very much, Rangarao, sir.
Can you make a full screen, sir? Can you make a full screen?
Yes, sir. I will.
Yes. Yes, thank you, sir. Thank you.
Yes, thank you very much, sir.
Please go ahead, sir.
Yeah, thank you very much, Rangarao, sir, and Jalja, madam, for inviting me to this seminar. And in fact, I am very much thankful to Dr. Prashant, who introduced me to this group. And Jagdeep Sohel. I think Jagdeep is not here. So, thank you very much. And my name is Rahul Narang. I am a medical microbiologist by profession and by training. But then over a period of time, we have been working with veterinary people, with veterinary microbiologists, veterinary public health. And then we started working with environmental people working on environment. And finally, we developed a center in our institute called One Health Research Center. And we were also participant for development of National Institute of One Health in Nagpur, which is just 80 kilometers from here. I am also part of Indian Association of Medical Microbiology, and I'm coordinator for their One Health working group. So what I'll be talking about is paradigm shift in infectious diseases, infectious disease laboratory system in India. Will this be new normal beyond COVID-19? So this presentation I had made in 2018, modified it in 2019, and now in 2020, whatever we had proposed that time has become true. So learning objectives for this presentation are, One Health concept, then zoonotic diseases of public health importance, which can be foodborne or RNA organisms, and then we are talking about RNA viruses and their emergence. What happens when they emerge or re-emerge? What are the responses? How can we predict and prevent impending outbreaks? And I'll also talk about our Indian Association of Medical Microbiologists' role in One Health and how role of clinical microbiologists has changed over time. So while I'm talking to your group, sir, so I may be little out of place, but then over a period of time, what we have learned that we should understand each and every discipline, and we should respect each and every discipline, and together we can collaborate and coordinate for various conditions, including this COVID-19. So health, if I'm a medical doctor by profession, so health for us as defined by WHO is a relative state in which one is able to function well physically, mentally, socially, and spiritually in order to express the full range of one's unique potentialities within the environment in which one is living. So that is health defined by WHO. And One Health is nothing but an approach. It's an approach to designing and implementing programs, policies, legislation, and research in which multiple sectors communicate and work together to achieve better public health outcomes. Again, defined by WHO in 2017. And One Health Commission has defined it as a collaborative effort of multiple disciplines working locally, nationally, and globally to address critical challenges and attain optimal health for people, domestic animals, wildlife, and our environment. So this definition, second definition, it fulfills all the criteria, and that's why we working in medical microbiology and public health microbiology, as well as animal microbiology and animal public health, together with environmental people, are working together on One Health. Why this One Health approach is required? Because we know that many of the microbes, they infect both animals as well as humans, because we are sharing the same ecosystem, and efforts by just one sector cannot prevent or eliminate the problem. So my previous two speakers, they have made my presentation very easy to understand, and I'll only talk about COVID little bit, and I'll talk about in general what we do when we have such outbreaks and when we are expecting such outbreaks and how can we prevent these outbreaks. Like I said, I made this presentation in 1819, so that time COVID was not there, but Nipah, we know that Nipah and Hendra viruses, both they have affected this whole area from Australia to Asia, including India. Initially started in proven that it can also be transmitted from bats' urine, which goes into palm sap, and ultimately when humans consume this palm sap, they get infected. Ebola virus outbreak has been going on, latest one since 2014 in Africa, but then it has remained there and has not traveled to rest of the world. Another organism which is very important from global health security, that is H5N1 or highly pathogenic avian influenza virus, which is also zoonotic disease and makes human population at risk. We also worked, while working on our project on zoonosis, so we tried to model for disease, not actual model, but we were trying, and then we put all those our findings and ultimately concluded that probably there will be an outbreak in one of the villages, and this outbreak happened. We predicted in 2016, and in 2017 this outbreak with leptospirosis happened in Wardha district. So that's about our work here with viruses as well as bacteria. So we are working with Japanese encephalitis and other viruses causing encephalitis in humans, and we are also working on HIV, that is part of the national program, and now we have COVID laboratory, and we also have COVID hospital in our campus. But then, and that is different. Now, what for today, in 2017, Global Health Alliance had given this map, and they had shown that China and India were the global hotspots. And then they had written that 1.5 million plus viruses, they are lurking in the various biodiverse regions, and they can come up anytime. And we have seen first example in the form of Nipah virus, and now we have COVID-19, and then Zika was there in initially in Latin America, but then in India also we have started finding these cases. So why India is more prone for viral diseases and other diseases of animal origin? So it needs population density. So now if we go by population density, then we have more than 1.35 billion people, 512 million livestock, and 729 million poultry. So the contact between human and human, animal and animal, as well as human and animal, is at very high rate. And this increases the potential for emergence, circulation, and sustenance of newer pathogens. Now, as India is progressing, we are also losing forest. Now the government is trying to make man-made forest, but the original forest which was there has been lost. And what has this done? It has brought wildlife near to livestock. So in this photograph, you see the livestock is grazing along with wildlife. So any disease from the wild can go to our livestock, and ultimately it can come back to human beings. We are performing a study on Zoonotic Tb in animal, human, and wildlife interface in Wardha district and adjoining district, and maybe after 1 year results will be out. Food can act as a source of outbreak. Now, some of you may not like this photograph, but then this is truth. People have been eating animals like anything, and some of them, they may not have access to other kind of food, like this rat eating community of Bihar, but then others who have well access to any kind of food, they also eat this raw food. This is photograph from Ethiopia. And then whenever animal, humans, they are in contact with each other, then viruses as well as bacterial. Say for example, this E. coli, which is say 0157, it is there in the healthy animals, can be shed by these healthy animals goes to lettuce, and ultimately when humans consume that lettuce, that goes into their intestine and can cause hemolytic uremic syndrome. Okay. Now, food has caused outbreaks. We talked about Nipah virus, which has been transmitted through date palm sap. Then Trypanosoma cruzi, we know that redovid bug is the causative agent, means transmitting agent, vector for this disease, but then people have also found it in sugarcane juice. And we are all, we know very well that listeriosis is associated with refrigerated food, raw food, both vegetables as well as fruit. Now, today we are focusing on COVID-19, so RNA viruses, and these are the cause of worry in recent scenario. That's what I had written in 2018. And there are around 180 currently recognized species of RNA viruses that can infect humans, belonging to 50 genera and 17 families. On an average, 2 species are added every year. They are routinely exchanged between human and other hosts, particularly other mammals and sometimes birds. We talked about zoonotic diseases, which are transmitted from vertebrate animals to human beings. So 89% of human infective species of RNA viruses, they are zoonotic, but then others will also have zoonotic origin. And this now working definition of zoonosis, it ignores arthropod vectors. So previously, if you remember, we talked about diseases as zoonosis that were transmitted by vertebrate animals to human beings. And that time, scrub typhus, which is caused by a bacterium Susugamushi, Orientia Susugamushi, was not considered as zoonosis. But now, later definition has ignored arthropod vectors, and they say that these are just transmission routes rather than alternative host species. If we are talking about viruses, then we have to think about various viruses and how they get transmitted to human beings. Initially, all these viruses, whether rabies or Ebola or dengue or HIV, they were in animals and they were being transmitted among animals. Later on, some of them, they caused primary infection in human beings. So rabies, if it causes infection in human beings, then human beings become dead host. No more further transmission. Second was this Ebola example we can take of this Ebola, where from animals it goes to humans, but then for some time it can be transmitted from human to human, limited outbreak. Now when this COVID-19 started, people thought that probably there will be limited outbreak, transmission, and then it will die, the disease will die, but that didn't happen. Then fourth stage is long outbreak, where the disease goes from animals to humans and ultimately starts transmitting among human beings. Like, say for example, for dengue, if the cycle is between animals, we call it like sylvatic cycle, and if it is among humans, from animals to humans, then humans, then urban cycle. So most of the time these days we have urban cycle of dengue going on. Fifth stage of a virus is when the virus becomes an exclusive human agent. HIV has become an exclusive human agent. COVID now has become an exclusive human agent where human to human transmission is going on. And now people are talking about community spread. The government agencies are not ready to accept that community spread is taking place, but examples show that there is community spread going on. Now, why do these outbreaks occur? These outbreaks means while the humans were living, animals were living, and they were living happily, everybody was living happily as the human evolution was taking place. But then that time, not many diseases we talked about. The reason could be that time not many diagnostic facilities were there. But still, slowly and steadily after this new century has come up, we are seeing more and more diseases. So climate change has affected environment of humans as well as animals and their vectors and pathogens infecting both. So we have found that there is a strict relationship between global climate change and the factors like, say, increased greenhouse gases that has led to reduction of coral reefs due to marine acidification and up to 40% reduction in oceanic phytoplanktons. Then global climate change has also evidenced that emergence of disease like dengue hemorrhagic fever, Rift Valley fever. Nowadays we are seeing this Kyasanur forest disease. Initially it was restricted to forest in Shimoga, but now it has gone to Maharashtra as well as Gujarat. This is one chart which I want to show in every presentation I make on One Health. And this is, we call it as One Health Cosmos, prepared by Z et al. See, what it shows that there is health and there is disease. And there is a fine balance between health and disease. And this balance is maintained by animals, food, our ecosystem, and global health and health policy and international health regulation and media and all these things. So whenever we have any disease which is coming up, then either it is food. Say we take example of COVID-19 or SARS coronavirus 2, whether it has come through food or it has come directly. I don't know. I don't want to comment about that. But then animals were involved. It's a zoonosis, and then human public health, education, biomedicine, microbiology, veterinary medicine, epidemiologists, vaccination, everybody has come into action. And so this whole thing will impact how the diseases, their presence will be lowered. Say for example, the blue lines, they show that the diseases will be less, because if the economy improves, there will be lesser zoonosis. If there are lesser animals, then there will be lesser zoonosis, something like that. But then biosecurity is another issue. Means this is a controversial issue how this virus emerged, but then biosecurity is another issue that we have to think about. Now, whenever these diseases, they emerge or re-emerge, then there are emergency responses. So emerging infections, emerging infections are described as newly identified zoonotic infections at animal-human interface. We talked about interface. And often they are first identified many years after the breach in species barrier had occurred. So same has happened with SARS coronavirus 2. And during past 40 years, newly emerging infections like SARS or SARS coronavirus 2, Ebola, Marburg hemorrhagic fever, HIV, Hendra, Nipah, we talked about them. and certain foodborne bacterial diseases have come like this. Most emerging infections have been first identified in humans before their animal source was known, so the same thing is happening. And many of them, they re-emerge when the risk factors for cross-species transmission align. They can cause asymptomatic or mild human illness. Now, we know that 90% of COVID-19 is people are infected are asymptomatic, or they suffer from mild disease, or they can cause severe disease. That's what my previous speaker was talking about, that there are certain predisposing conditions which will lead to severe illness, and sometimes healthy people can also suffer from disease. Now, since we work with the IDSP, that is Integrated Disease Surveillance Program, and International Health Regulation, so I'll talk about this little bit. So whenever there is an outbreak, what emergency measures we take? And after this outbreak, now this is pandemic with COVID-19, most of you already know what we do. So first of all is epidemiological investigations. They are initiated, and we look for risk to humans, and we knew that the risk to human with COVID-19 was more, especially in Western world. And then what is the source of the organism? My previous speakers, they have shown us how we can go to the source, and if it's animal, then whether there is continued risk to humans by presence of animals. We are talking about those diseases which came to humans and then got suppressed. But this COVID-19 is still continuing between human to human spread. What emergency measures we take? We start doing surveillance. Now, latest is ICMR is doing surveillance, has tested 26,000 people for IgG, and have shown, I mean, we will not talk about those results. And then contact tracing, all those who came in, were positive for any disease, including COVID-19, then their contacts are traced, and then we look for disease in them also. Then isolation, now people are well aware, isolation, quarantine, social distancing, vaccination. Still there is no vaccine. What we have started talking about vaccines, prophylaxis, and culling of the animal source if it has been found. This revised international health regulation has asked member states, or this WHO member states, to rapidly assess an emerging infectious disease outbreak. Now, again, it's a controversial issue between US and China and other countries, whether China should have informed beforehand. We will also not talk about that. But then all states, they are member states, they are have to notify WHO, and through WHO, the global community, if there is a risk for international spread. So since there was a risk for international spread, this should have been done earlier. And what are, I'm sorry for all this, what are the economic impact of outbreaks? This is a healthcare and public health response, and reduced tourism and travel, trade restriction, reactive animal control strategies, say for example culling, and we have to do compensation also, and workforce absence. All this we have seen with COVID-19. Now, why we are talking about One Health? And so there has to be a shift in paradigm. Now, initially, it was like pathogen, it circulated in animal host, then emerges in humans, and infection and disease occurs in human beings. Paradigm shift should be, if we find disease in humans, we should try to prevent its transmission and predict its either emergence or similar disease emergence or re-emergence, and we should try to control in animal population wherever feasible. Say, we had MERS, we had SARS, and SARS probably led to SARS coronavirus 2. Now, I don't know if we could have predicted it beforehand and prevented it, but then it has happened. So how does this One Health approach work? We have to predict and prevent the disease. So we have to prevent human infection by enhanced detection and surveillance in both wild as well as domestic animals. And such geographical localization is followed by a prediction of which organisms might emerge based on understanding of what infectious organisms are present in wild and domestic animal population at those sites which we look for. Now, I'll give you an example of zoonotic TB. See, for many years, we believed that Mycobacterium bovis is cause of zoonotic TB, and we always reported Mycobacterium bovis. Now, a recent study which has been published in Lancet Infectious Diseases just 3 days back from CNR, they have tested around 700 isolates of Mycobacterium tuberculosis complex, and they did not find any single Mycobacterium bovis in human beings as a cause of zoonotic tuberculosis. Instead, what they found was Mycobacterium aerugis. Now, the tests which we perform for Mycobacterium to differentiate between bovis and Mycobacterium tuberculosis, they actually do not differentiate between Mycobacterium aerugis and Mycobacterium bovis. So now looking at taking into account all those results, we are planning a study in our animal, human and wildlife interface if aerugis or bovis is lurking in our area and whether it will affect our human population if we try control Mycobacterium tuberculosis by government efforts. Now, then I'm thankful to my previous speaker. They have already talked about genetic sequencing, and so sequencing is important. And then this One Health approach allows development of upstream approaches to predict outbreaks, that the paradigm shift we talked about, and build upstream barriers for transmission. That's what we are trying to do with TB. And this approach will demand close collaboration among environmental scientists, veterinarians, physicians, coupled with computational scientists, mathematicians who can build powerful predictive models of disease transmission. Now, for some time, we have been talking about these models, and now I know that among this group, we have computational scientists. So I'm very lucky to be part of your group. We can say now our group, because we have been working with veterinarians and other people, but then we did not have much connection with computational scientists and other people working in the field of science and human pathogens. And maybe in future we will be able to work together. Metagenomics means for us as a medical microbiologist, it was something that I had to teach those students, but then today we will not talk about metagenomics, but then you already understand that metagenomic studies can tell us which organisms they are lurking in the environment, and if the environment changes, then whether those organisms can cause disease in human beings. And it can also tell us the virulence genes within that area. And people have also sequenced phages. And this data of metagenomic can identify reservoirs of pathogens in an environment and predict environments with a high probability for evolution of new pathogens or outbreaks caused by known pathogens. So this is what my friend in AIIMS Nagpur and I, along with National Institute of One Health, we are working on, and we will need help from your group, sir, to develop a model, and we should be able to predict such diseases at least maybe 5 years down the line. And okay, we will leave this. Okay, now One Health approach is coordinated by WHO along with organization, this Food and Agriculture Organization, FAO, and OIE, that is World Organization for Animal Health. And so we have already talked about various sectors working together. What is the Indian scenario? Means in West, people have been talking about One Health since long time, say 15 years. So and now I'm proud that since 2015, we had been working on a project, task force project, joint project of ICMR, ICAR, working on zoonosis in central India, and that has now led to setting up of National Institute of One Health. Initially, it was to be called National Institute for Zoonosis. It is run by NIV, and MAFSU is Maharashtra Agriculture University in Nagpur, and National Institute of Epidemiology, MGMS, is partner to that. And there are many other agencies who are working on One Health in India. Kerala, we have a Center for One Health Education, Advocacy and Research, and now many universities have started establishing this center. This was the first center in India. Indian Association of Medical Microbiologists, they are working in 2016. We established our One Health working group that was to work on zoonosis, laboratory strengthening, and biorisk management for Indian laboratories. So we started working on laboratory mapping, human laboratory mapping and strengthening project along with National Centers for Disease Control, and we surveyed different states including Tamil Nadu, Gujarat, Jharkhand, MP, and now government is working on their strengthening on including molecular assays, quality management system training, biorisk management training, etc. And see how this program has helped. So recently, Medical Council of India, now that's no more, so Board of Governors, they have said that each and every medical college should have a molecular biology laboratory. And all the medical colleges, are already working on COVID for this laboratory. So lab strengthening has actually helped. And then biorisk management. People talk about Wuhan laboratory and then biorisk management in that laboratory. So since 2016, we have been working with this one agency in US where we are training microbiologists from different streams on biorisk management. And then we also had this meeting between different partners for strengthening preparedness for emerging and re-emerging zoonotic diseases. That was in 2018. And then we also conducted tabletop exercise on One Health. Again in 2018, India had One Health Summit in 2018. So 2017, 16, 17 onwards, people have been talking about One Health and various diseases. This is the area where we work in central India. This is that time DG ICMR, now DDG WHO. So she also came to visit our area of work on zoonosis. And we established One Health Containment Laboratory. In 2016, we worked on viruses, bacteria, and a lot many other diseases, and see how it has helped. The moment government asked us to make it an exclusive COVID-19 laboratory, we just needed from government the kits. So what we propose now that each and every medical college and veterinary college and other colleges should establish such laboratory. That should be paradigm shift for everybody. But then since I belong to medical microbiology, I can talk about my discipline for the time being. And all these, we have different branches of microbiology, and medical microbiology is just one small branch of microbiology. What we have been doing all the time since long, we were restricting ourselves to laboratory diagnosis, and that was our conventional role. We were working on infectious diseases. Then antimicrobial resistance came and we started working on it. And slowly and steadily, we said that now we are clinical microbiologists. So from medical microbiologists, we became clinical microbiologists. We started interacting with clinicians on various conditions of the patient. But that is one aspect. What I want to emphasize is we have to be community microbiologists. If we have to talk about various diseases which are lurking in the environment, which can cause disease to human beings, microbiologists, medical microbiologists have also go and play a role of community microbiologists. And I talked about IDSP, which can predict infectious disease outbreaks and give some data to the government to work on it. So this diagram I have been showing for last 3 years, and I say that as a medical microbiologist, we have to look at the penumbra. We have been looking at this umbra for a long time, doing conventional microbiology, quality management, biorisk management, but then all these proteomics and genomics, we never paid much attention to them. But now slowly and steadily, we have to talk about them. We have to talk about metagenomics, we have to talk about next generation sequencing, and we have to talk about public health microbiology and ultimately One Health microbiology. So who brings this change in us? The organisms, they bring change in us. So conclusions, the emerging and re-emerging diseases will keep on raising their head. We have to strengthen preparedness, and that is the need of our regular interaction among experts from public health and microbiology laboratories, human and veterinary, and now after this I have to add computational biology and other laboratories, and that should become norm these days and beyond COVID-19. IMM has already taken steps keeping pace with other national and international agencies, and some of us, that time I wrote that some of us medical microbiologists have to accept new role as One Health microbiologists. So thank you very much, and I acknowledge IAMM, ICMR who paid for our studies, and my institution, MGMS. Thank you very much, and I'm sorry for this long presentation.
Thank you very much, Professor Rahul Naran, sir.
Hello.
And highlighting about One Health approach and comprehensive understanding on zoonotic disease and their transmission, and also importance of RNA viruses causing disease, and then emerging measurements, outbreaks, and economic importance of outbreaks, and metagenomic approaches, and role of IAMM in One Health approach, and then shift the paradigm, emerging response to preventing emergency at the sources. So I am very happy, sir, and hope everybody enjoyed this presentation. And now actually open for the discussion. So you are welcome to participate, ask questions our speaker.
Hello.
Yeah, please.
Hello.
Hello.
This is Professor Mainos.
Rahul sir, can you hear me? Can
Yes,
you hear?
sir.
Yeah,
Yes.
okay. Yeah, please go ahead. Hello.
Yes,
Hello.
sir.
Can I have one query with Dr. Rahul Narang, sir?
Can you speak loudly, sir?
Yeah. Hello.
Yes, sir. Please.
Can you hear me?
I'm hearing.
There is one query for you, sir, with regard to this. I'm Professor Bharatkumar from Rayalaseema University, Kannur. We have gone through last 80 days around lockdown 1, 2, 3, 4, and the fifth one is going. To compare them, the previous lockdown process is somehow relaxation has been there. And moreover, the people, they have been taking care by their self and all without having any kind of what you call the protection measures and all. So in the part of this sort of scenario, what kind of what you call immuno boosters or the supplementary systems they may supposed to go for? Is it required? Everyone should have to go for while using N95 masks or any other what you call sanitizers. They will also clear out the used bacteria from the surface system, hands and all. Is there any other things we can able to go for to overcome this sort of scenario or this situation? Please.
Sir, means it's a very difficult question actually to answer.
Sir, please.
The thing is, the only thing as a medical microbiologist which I can tell is recently it has been said by CDC that this virus can be transmitted from human to human by airborne route. Most of the time we were believing that it is droplet nuclei which will droplets which will settle and then humans will get infected. But now we know that transmission is going on by speaking, even by breathing, the aerosols can be generated. But then when the person sneezes or coughs, then these aerosols go a long way and they can infect. So if that is the case, then wearing masks becomes mandatory. WHO sometimes says and makes conflicting statements that it may spread, it may not spread. But then I personally believe that wearing masks while you are in the community, if you are sitting alone in a room, driving your own car, then it may not be required. If you are walking on the road alone, it may not be required. But while you are along with other people, you have to wear that, sir. And second thing, I believe that sanitizers means our hands keep on becoming dirty, so sanitizers are important. But then as a medical microbiologist, I am used to washing my hands regularly with soap and water, so I feel that is the best thing that can happen if we have access to soap and water. Now, second part of your question, sir, immunity boosters. So Ayush Ministry has given guidelines and they have said that these should boost the immune system. And I have been talking with Dr. Kavi Kishore sir and others regarding other immunity boosters, including curcumin. And means that's my personal view and sir's view also, so that curcumin can also act as an immunity booster. But then Ayush Ministry, I don't know why they didn't allow it, but it's a very difficult question, sir. It means if you feel you take, but then means I can't answer that question, sir. Regarding hand washing and mask, I can answer.
Yeah. Sir, thank you very much, sir. Your lecture is very informative. Is the future to call object results has been referred in a very good way. Thank you once again.
Sir, Professor Rahul Narang, sir. Sir, there is one participant, they are asking questions, sir, one question.
Yes, sir.
That is, will the virus be in a sleeping stage in an infected person when he returns negative? If not, why there are cases which are turning positive again from negative?
Sir, yes, sir. That is pertinent question, sir. Now you see, that ship, no, diamond ship, where this virus was seen, and so that time, even if people became were not coughing, the virus was there in their intestine, And later studies, they prove that virus, although is there in the respiratory tract, will also be there in the intestine, and in the intestine it will remain there for the next 10, 11 days. Question arises, two questions the person has asked. One, after the person becomes negative, then whether the virus will be in sleeping stage. Now, what we are doing to detect this virus in respiratory samples is real-time PCR, and that has got a detection limit. And various, this question also I have been discussing in this group, that some companies, they say that CT value of 40 is the cutoff point, the ICMR kit says 36 is the cutoff point, and but then ultimately while it goes towards 36 or 40, then the virus is there in the respiratory tract, but then it is to such a low extent that while the person coughs or sneezes, then it doesn't get carried to the other person. So the risk of transmission is low in the respiratory tract when we say the person has turned negative, even though virus will be there in the respiratory tract. And then virus RNA has been found in the intestine. And now whether the presence of that virus in the intestine leads to transmission of disease, Dharavi outbreak could be one example where if it is there in the intestine, it can roam in the community, but then transmission through respiratory route is more likely. But virus is still there while we say the negative, and then studies have proven, sir, the person doesn't become positive again. Means there were reports from Korea, and in our hospital also, we had one person where the report came negative and then came positive, and that was only because of the cutoff titer or cutoff CT value of the kit. So people, they were using a kit which had mentioned that cutoff was CT 40, but the value of 36 was given as negative. And later on, somebody else reported with the same kit, and he said, no, this is positive. So that means person becoming positive again is very difficult to say. Means it has not been proven, sir.
Okay, thank you very much, sir. Thank you very much again on behalf of our this one. We are very much thankful to you, sir, for a very informative lecture. So now we will move on next speaker, sir. Thank you very much, sir.
Thank you.
So can you make a host, sir, if you don't mind, Professor Rahul, sir?
Yes, sir.
Go to participants. Go to participants.
What more? Okay. Participants.
And right side more is there, sir. You click your name and more is there.
Just a minute.
Or you can click on my name, sir. Click on my name and host. More it is there, more, and then right click, make host.
Just a minute, means I am finding it difficult now.
Yeah.
I'm sorry, but
can you click on my name, sir? Can you click on my name, Rangarao? Find a participant. You type, sir. Find a participant.
Okay. Yeah.
Rangarao.
Yes, sir.
Rangarao.
Yes, sir.
Right. Just right side. You can see, sir. Mute and more is there. Mute and more. You click more.
Yes, sir.
Right click. You will get make host.
Done, sir.
Thank you very much, sir. Thanks a lot.
Yes, sir. Thank you, sir.
Now move on next speaker. Professor Dr. Prashant N. Survajola, Senior Scientist, Department of Biotechnology and Bioinformatics, Belonged of Scientific Research, Jaipur, India.
Yeah, thank you, Dr. Rao. I think you have already introduced me. I think.
Yeah, no. We are not going to this one. Just, sir, I will make you host, sir.
Yeah, is my voice clear?
Yeah, yes. It is clear, sir. Please.
Can I share the screen, please? Yeah.
Yeah. Wait. Sir, I will make you host.
Yeah.
Yes, sir. I make you already done, sir. Please go ahead, sir.
Yeah. So I think I hope you can see the slides.
Yeah, definitely. Yeah, sure, sir.
Yeah.
We can able to see.
Yeah, thank you so much, for this gracious invite again, Dr. Rao.
Yeah, you are welcome,
I thank my mentor, Professor Kavikishore, sir, and Dr. Jalaja as well, for the kind invitation. It's been absolutely an amazing talks, the last, 3 occasions, right from Professor Krupanidhi, Obul Reddy sir, who was my senior, and of course, Dr. Rao Naran sir, who has given wonderful talk on One Health. So my talk is on systems challenges of COVID-19. So I come from Birla Institute of Scientific Research. We have got state-of-the-art laboratories, and we have got 2 main departments and about 14 scientists. We are here with about 7 PhD fellows and 2 postdocs. So my talk, probably I'll split my talk into 3 different stories. So one is on the history and the kind of pathogenesis, and then I'll slightly move ahead, giving some kind of problems and challenges from the phylogeny that we have done. And I'll also try to describe the pathway tools for metabolism. And the most important part of my talk is curcumin, which was proposed by Professor Kavikishore sir and Professor Giri sir, which allowed us to work on docking done by Abhinav and Renuka. And then finally, I'll just try to conclude with some perspectives. So this is what I've written an article, sitting at home called the 3 C's. One is collaboration, convergence, and consistency. And incidentally, I'm really honored today that today's Science Edition editorial, has specifically mentioned convergence, the role of convergence for the scientists, because we're all divided, by nations, divided by labs, but we're not really, trying to converge here. We're not really try to bring up open collaborations, which is really a farce, which is really very unfortunate. So if we can bring together collaboration, convergence, open up some ideas, share some research spectrum, and if this continues, that would really bring out good consistency against this human activity of the crisis. So this is what I was just trying to tell you. So how do we make our environment safer? So there's a release of greenhouse gases, and this wonderful article by Manuel et al. In 2020. It was published a couple of weeks ago. This gives a wonderful explanation, how best we could stay home, and we have been reaping results the last 3-4 times. So as humans, we are just we need to be a bit selfish. That selfishness is like we could save energies, we can switch off the vehicles at the red light traffic zones. Probably we can make our lab and working space clean, and most importantly to the young generation, let's clean our phones as well, which has a lot of crap these days. That will really probably allow us to encounter the future next big data challenge, because according to Moore's law, the computational space needs to be doubled as the value of that particular price goes down, or to a half. But the take-home message over here is, what I really wanted to tell you is, because of the temperature, the virus has an ability to probably something different when compared to the previous cases like MERS or the other pathogenic cases, where we are just beginning to understand why is that even at a very extreme temperature, even at 40, 45, the survivability rate of virus is really extreme. Why? So this particular question has got some meaning here. So to understand this, we cannot really directly experiment on the humans at this point of time. So we are trying to attempt some kind of models using systems approach, which we call it as complex pathway simulation model. So we create a kind of a virtual cell and try to ask that particular virtual cell just like to behave just like a human cell, and give the extreme temperature to that particular cell, and we try to induce some virtual viruses or bacteria into that and see how the virus fares or how the virus behaves. So this is what we are just trying to understand. And if you see, these are all the keywords I think, SARS-CoV-2 is really affecting every disease, whatnot. There are a lot of comorbidities associated with that. There are diagnostic front, coming from QRT-PCR, laboratory diagnostic kits. There's antibodies, like we are working on aptamers as small molecules, with the Greaser and Kavikishore sir. So all these kind of things are really coming up. At the same time, there's also vitamin M, vitamin money. So the onus is on we humans to come up, wonderfully, come together. It's not really at stake. And for that, we just need to bring together all possible collaborations, to the fore. So this is absolutely, very, very, what probably I would ask is, if there are so many mutations that are arising, not 5% unique mutations across the entire spectrum. So we just, try to ask, why is this? If these particular mutations are trying to bring out any non-synonymous change into the viruses, thus behaving or thus bringing up a lot of, novel proteins. So just like a human host, we also have a systems approach. This systems approach is in such a way that just because of that change in the mutations, we might really come up with a lot of, cells, and as a result, the entire pathways, might really change. So this is where, the strains, try to adapt, and probably they try to de-adapt or unfit in the environment. And probably, as a systems biologist, I would probably argue that, the SARS-CoV-2, although it's might, it's really, behaving, very rapidly, and there's a likely chance that, it might even remain in the next couple of years as well. So, story is, I hope I'm audible. I'm sorry, I just wanted to just make sure, like,
we can hear you.
Dr. Rao and others. Yeah, thank you so much. So, so where are we now? So, if you ask, the known unknowns of COVID-19, there are different number of bioinformatics tools. Those are listed on this particular page, as you could see. So, they're produced from GISAD, which is a global initiative for all influenza diseases. 9,000 assemblies of SARS-CoV-2 are listed, out of which about 683 as on today are from India SARS-CoV-2 genomes. So as you could see from the slide, there are different soaps simply labeled as G, GH, GR, L, O, S, and V. And as you could see, the majority of them from North America are from GH. And of course, if you really see from Asia, it's really, from the O clade, which is, the O strain. And as from the previous speakers, from wonderful lectures, I think, we obtained it from partly, from, Europe. And this is how all, phylogenetic tree that's being really updated, from time to time. The predominant clade, all the red ones are all the strains that have evolved from Europe. Okay, and the blue ones, as you could see here, the blue ones, they're seen across all the different ecotypes, and they're all, from the Asia Pacific regions, and the majority of them are really mild, and they're really coming from India. So, when this SARS-CoV-2 was into picture, I also have become an opportunist in the form of an opportune virologist. Although I'm a biologist, I didn't work on viruses at all earlier. But as an opportunist, we just try to explore. And from the umpteen sequences, about 683 sequences from India alone, a lot of them are all having ends, which means the sequencer, couldn't read what that particular base is. So that is what we call it as a noise. So around 72 India SARS-CoV-2 genomes, unfortunately, have so many stretches. So which means sort of around 600. So which is which is about 1/5 of these particular sequences are really MS. So this is because with due respect to all the librarians or sequence librarians, there's a big mess going on over here. They need to be assembled properly. And many of the samples could not detect NSP7, which is a non-structural protein 7E and 7B. And if you ask me, it's a bit kind of a tricky situation. If we have a viral load inside the host, and if you take a good CT value of anywhere between 1 to 15, which means if the CT value is less, then the viral counts could be pretty higher. What should be an ideal read depth for this? So for a sequencing chemistry of 2/150 base pair chemistry, an ideal probably for meta-next generation sequencing could be 100 million reads. But if you're really looking for the comorbidities, which means for the samples, probably you may really want to check if those particular samples have got any other diseases like diabetes, hypertension, and other stuff. Maybe you could even still come down to about 5 million rates as long as you check the comorbidity list. So SARS-CoV-2 has become a diagnostic teaser ever since it was reported in December 2019. So as I told you, over 39,000 assemblies are there, and about 4,000 are from China and Southeast Asia alone, and about as on today, this slide was made, yesterday morning, and during the last probably 24 hours, another 80 sequences now cropped up from India. So there are about 595 sequences. And usually the samples that are sequenced are nasopharyngeal swab samples, esophageal or oropharyngeal samples, sputum, and of course, the valve, and in a rare case, the blood, because the transmission is not really through blood. So there's a difference, as I told you, between, as the speakers have wonderfully expressed. Novel coronavirus or SARS-CoV-2 clades differently from SARS-CoV-1 and MERS. I'll probably skip this slide, but probably what I'm just trying to tell you at this point of time here is that there are spike and nucleocapsid proteins which play a very important role, and their structures have really been deciphered. So we have got 4 structural proteins abbreviated as MENS, the membrane, envelope, nucleocapsid, and the spike. There are also other proteins, the non-structural proteins like NSP1 to 10 and also NSP 11 to 16 also do exist, but they're not properly annotated. And a big one, ORF1AB, which is a polyprotein. So, to our understanding, or particular directions of the last several weeks, so novel coronavirus clades similarly and yet differently. So when I say similarly, so this is the SARS-CoV-2. As you could see, it clades with several nearest neighboring sequences. Many of them are SARS-CoV-1 and also SARS-CoV-2 pathogenic strains, those 4 HCOV strains that were reported earlier. And it also has a distant correlation with the bats over here, where the horseshoe bats were the possible reservoirs. And from this particular slide, as you could see here, the blue clades, these are all from the Indian perspective. So as you could see here, the Indian originated clades have been distantly placed over here. So say for example, here, as you could see, the red one is absolutely pathogenic, but all the blue ones are absolutely mild, which means they're not that friendly clades. So, so you have something like kind of a heat map across all the continental sequences, so from Asia, America as well. And we just try to see how these particular sequences are similar. So in conclusion, the take-home message from this particular slide is all SARS-CoV-2 sequences are 99.3% similar, which is minimum. So which means there are about 0.05% or less than 0.05%, unique mutations that are heralded with SARS-CoV-2, and that makes pretty distinct. And those mutations, really make sense for development of diagnostic therapies. A beautiful picture came into being in 2020, where Hamad et al. Have published. So they have deciphered the sequence identity and the structural correlation using the Circos phylogram of all the structural proteins, and they just tried to attempt the genetic clades and how they are really outgrouped. So keeping that into picture, what we did was we have taken the entire 29.9 kb region of the sequence, which is a SARS-CoV-2 sequence. We asked how many of these particular sequences have got the open surfaces, which means there are some exposed surfaces in the form of amino acids of the entire proteome of SARS-CoV-2. And if those exposed surface regions form a part of the helix, and if they really form a part of the helix, would there be any epitopes tagged to that? So if there are epitopes and if there are exposed surfaces, if there are the structure is the helix, then that indicates that those particular regions could be better targeted. So as I worried. And interestingly, these are not, these are not, tandem. As you could see, are different, like 1202 to 1204, you've got Kia amino acids. One is exposed, the other one is buried. And of course, here from 1275 to 1279, there are buried and exposed surfaces. So to identify probably a good candidate, these make sense. And usually people only go for the 4 structural proteins, the nucleocapsid and other stuff, but we also identified some of the proteins like ORF7A. The reason why I'll probably tell you in the next few slides. So the second part of my particular talk would be about sequence variation, which we have done an exciting work with in collaboration with Keshav Singh. Professor Keshav asked was if there is the entire length of the sequence, if there are any functions that we could really do sequences, how these particular sequences vary across different continents. So if at all, if they really vary, could epitopes be that could also be varying? Because the end goal at this point of time is we might really go for something called aptamer-based testing, where aptamers as small molecules could probably replace antibodies. The reason why I'm my first postdoc in 2010, I extensively, and we used to pay about $10 for an aptamer, be it a DNA aptamer or an RNA aptamer or even protein aptamer. But now the same aptamer costs about $450. We try out a signal from a Western blot antibody mapping technology. Probably you could probably with an aptamer with highest specificity and probably better sensitivity. But now you see how the pharmaceutical companies are really making a big market taking the aptamers. So the next evolving story. This is a wonderful collaboration from Professor Keshav Singh and Gyaneshwar Chaudhary from Binaras University. Yeah, hello. I hope I'm audible. I'm sorry. Yeah.
Yeah, we can hear you.
Yeah, sorry, yeah. So, what we asked was, we have taken the entire SARS-CoV-1 and SARS-CoV-2 sequences. We asked, what is the probably ORF3b is present and ORF3b is absent in SARS-CoV-2. ORF3b is a protein that is absolutely targeting the host cells and probably targeting the mitochondrial sequences. So what happens is, when SARS-CoV-2 infection takes place, it enters the host. So if there are usually a healthy cell usually has anywhere between 400 to 2000 mitochondria per cell, which allows us to respire. This guy does is, this particular SARS-CoV-2 goes inside the host cell, absorbs the mitochondria, ensures that its erstwhile derogatory ORF3b is completely gone, which means the entire SARS-CoV-1, 2 has evolved to SARS-CoV-2 at some point of time during natural selection. And then the entire ORF3b is depleted now, and that is absolutely lost, where it is really integrated with the mitochondrial DNA of the host. So if the entire mitochondria is lost inside the cell, then you have a lot of asphyxiating cells. So when we say asphyxiating cells, we've got a lot of breathlessness, and that is how the mild respiratory syndrome and all those kind of things occurs, and as a result, the patient really dies. Okay, this is really an unfortunate thing. So what we try to do is, as you could see the extreme leftmost side, this is COVID-1 from Wuhan. And we did COVID-2 Wuhan, we just try to compare this. And then we just try to ask how this COVID-1 differs from COVID-2 across all the continents. As you could see, we have got Korean, we have got South African, we have got Americas, we have got Brazil, we have got Spain. When this paper came into picture, we didn't have Australian thing. That's only a sequence. Yeah. That was the only sequence that we were probably missing. And that really allowed us to probably look into those particular possible insights, where we just try to ask, what are those non-consensus sequences across all these sequences? When I say non-consensus, they are non-repetitive sequences, which means they're absolutely unique. So all these guys are absolutely unique. As you could see, all the colored ones, these are all the unique. So these can be the better unique sites that we could probably look into this. And we got signature sequences like AD and K in case of ORF7E. In case of ORF7, we have got LEL, QD kind of things. In case of ORF8, we have got CXE motifs, which we have identified, and all these particular motifs, which we have really identified. And now comes a question, how these particular sequences make a ORF7A? Do they really make any transitions inside the host? How do they really interact inside the host? Do they really try to, do they really try to integrate into the host proteins and try to interact with them? And if so, how would the line-based studies have been underway in University of Alabama? So we've got some possible stories about the drug candidates, the efficacies and the pathways. This is where we have used the visualizers. The most important thing which Abhinav Parashar of Vignan University and Renuka Sulavajula, my wife, who has done this particular work under the directions of Professor Kavikishore and Professor Girish sir. So this is one of the highly downloaded articles, and thanks to the first two others who have really made tremendous efforts. I'm just trying to tell you the story over here, that we have taken all the SARS-CoV-2 structural and non-structural proteins, tried to go for plausible drug targets, the simplest Autodoc-based model using Vignan and Pymol. And then we try to check the antiviral mechanisms, taking curcumin as a plausible candidate and comparing curcumin with remdesivir. We have got several other drugs like ivermectin, Retrovir, azithromycin, and all those kind of things. So the take-home message from this particular slide is curcumin wonderfully fares well with all these particular proteins. With those, the least, concentration required, for ensuring that it would really be an affable drug target. And the story evolved in such is obtained from turmeric, which we call it as haldi in Hindi or paspu in Telugu. The bioavailability is always in question. So far, right from artemisinin, Professor Tutu, who is a Chinese Nobel laureate last year, who has wonderfully exploited works on artemisinin and as well as Chinese herbal drugs. She also worked on curcumin. Professor GP, Professor Kavikishore Sirs and Professor Giris's collaborator, he has also worked on curcumin. The bioavailability was always under question. But here, what all we need is a minimal concentration for this particular curcumin to exploit as better drug targets. And the pathway analysis is the nucleocapsid protein has got some interleukin 17A as a possible candidate where pathways could be explored. So in conclusion, when curcumin is taken, it plays a very important role taking the receptor targets. It would also consider the IL-17A as master regulator because IL-17A is a direct homologue of nucleocapsid protein, and it interacts with several other proteins, and they're localized to the same system as well. That's very important. So this is only a kind of a plausible theory, that we are trying to come up with this, and we have made that particular story, which we have communicated it. Of course, the prophylactic studies and the cell line-based studies, Professor Kavikishore sir is doing, and we're all working towards major paper. So the final part of the talk, the overall conclusions here and the challenges here is we need to bring a better understanding of epitopes and antigenic determining sites. That's very, very important. If we could be able to do this, then we are very near to the identification of drug targets, and that is where the convergence really comes into picture. I cannot work in isolation. I can say that I've identified this thing, or make kisi ko nahi bolunga. I'll do my own. That probably doesn't apply when we are really facing this particular crisis. Probably we need to come together. We need to share ideas. We need to probably take or share those intellectual property rights, come together for development of these kind of things. And of course, annotation and curation of better antibody targets or aptamers. We need more raw sequences. The current 685 sequences from India genomes is not enough. This is a mild strain. It's evolving. It's really evolving rapidly. We need at least I would say at least 10,000 sequences from India alone to identify this particular virus behaves. So we need a better understanding. So that's where the sequence variability could be questioned. Questions like you might ask, what are the thoughts on the nature of novel coronavirus transmission from animals to humans? Of course, earlier people have answered like the lateral chain transfer could also be the key. This is what I really believe. A very important question probably you might really want to be answered with virus and from virus. So that's where we need to make a good habit to live with it. And that's where probably we can come up with paper-based kits or simplest pregnancy-based kind of kits, which Professor Giris sir and Professor Kavikishore sir have developed in lab, and they're going for the patent from their major brands. But as a systems biologist, once we get these particular sequences, and that's where the machine learning heuristics come into picture. So we can train a network of these particular. Look, in the near future, there might be a new viral strain that is coming up, and by then a drug target. That's where is really the key essence for this. And my sincere request to all of you is to educate masses. As scientists, we need to educate masses. We need to ensure that our world is safe, and if the world could be safe, that is only because through our word of mouth. Not just the doctors, not alone can do all these kind of things. Big salutations to doctors like Dr. Rahul who have been doing human service, and wonderful collaborations from Professor Kavikishore sir, wonderfully exploited by Rinka and Abhinav. Thanks to them, it was a wonderful learning experience from the functional biology side. And from the medical genomics, wonderful collaborations. In fact, I got in touch with Keshav Singh for almost 15 years. He was my PhD co-advisor when I started my PhD way back in 2010, financing the University of All India Institute of Medical Sciences. I'm the founder of this India's largest bioinformatics society. So please join GH, where one postdoc and 3 PhD fellows are working with me. And you can contact me at this email. Finally, I thank Professor Krupanidhi, Professor Rangarao, guiding for more than a fatherly like figure to us. He has been with Dr. Jalaja, Dr. Anil, and all others. Thank you so much. I'll be.
I have a question, Dr. Prashant. The ORF3b, do you think it has been deleted?
I would say it's natural only. So it might have. Might have happened, sir. But as humans, we're all scientists, and I would say we should be ethics because unless and until we do these particular experiments, I say this. But what might have happened? The hypothesis or theory over here is after going to so many sequences, they might be working with SARS-CoV-1 drug, and they were probably trying later attempts on the human clinical design, and incidentally that SARS-CoV-2 might have evolved from that bat-eating person who could really probably be a doctor or even could be a victim. So this is probably a kind of a theory. This is really quite amazing that ORF3b is the culprit for the mitochondrial this thing, because it also affects ROS pathway, the reactive oxygen species, and the entire mitochondrion. I mean, you'll be surprised to know that as soon as SARS-CoV-2 infects, depleted in a matter of minutes. So if there are 2,000 mitochondria in a healthy cell, it would really drastically come down to only 10 or 15, and that becomes mass fixating cells. So at this point of time, probably I'll be waiting, and that as a result, SARS-CoV-2 might have evolved, but probably that is not not done purposely. That is what I would like to comment.
Yeah, ORF3b stretch of sequence that is making this sequences.
Sir, there is also ORF7b, sir.
Both of them.
7b is also there. Recent sequence variation that we are trying to understand. If you see all the India genomes, especially the mild strains, NSP7A and across the entire SARS-CoV-2, especially from Indian conditions. So that begs us a question, probably ruled out, not just for ORF7A and ORF3b, or ORF7, 7A and 3b, but also for ORF8 and also NSP7A and NSP7b. So these are all the...
Yeah, out of the 4 different categories, COVID-19, protein do you think is more important as far as, containing the initial infections?
Right, I mean, the non-structural protein 10.
Okay, 10 is more important.
So that's because, sir, it has the most unique from our understanding, unlike the other structural proteins and other ORF for the non-structural proteins. So if there are better ways of ISIS are absolutely clearing these kind of proteins, that would really allow us to bring up some kind of theories in a better way. So I think NSP10 is the best, that is followed by ORF7E, followed by the structural proteins.
Is there any evidence that is being gathered, whether it is being inherited or not, we do not know at the moment. So is there any evidence that is being gathered in this direction?
There are certain theories here. I mean, for example, I would probably look into those particular patients because as of now it is seen only in males, because AST expression is seen more in males in terms of the testosterone. That's where a lot of comorbidities also do exist. To answer your first part of the question, we need comorbid who are not associated with other diseases, and just try to screen those particulars only with SARS-CoV-2 alone, who are young, age group between 35 to 45 probably. That would be an ideal one. So if there are some kind of ideas. So this is what...
Yeah, excellent talk, Dr. Prashant. I congratulate you. Thank you very much, for enlightening us.
Yeah, thank you, sir. Thank you so much for the kind invitation. Yeah. Yes, I think here, Dr. Rao, I mean, are there any questions from the participants? I don't see any questions from the chat box at least. Microphone to you, or can I make you the host?
Yeah, please.
Yes. Yeah, thank you so much.
Yeah, please. Thank you. Yeah. Now, actually open for the discussion. If participant would like to ask any questions, please welcome you all. You can ask any questions, Dr. Prashant. If nobody is asking questions, then now actually I would request Dr. J.R. Ravishankar, Professor Ravishankar from Bangalore. Sir, can you hear me, sir? Professor Ravishankar, sir.
Yeah, I can hear you.
Yeah, could you please share your views about this webinar? Sir, you are welcome, sir.
Thank you very much, Dr. Rangarao. Thanks, Dr. Kavi Kishore, for inviting me to this webinar, and it's been a wonderful selection of topics and wonderful speakers who are very efficient in each one of the areas which they dealt with. So to begin with, the Krupanidhi's talk was very interesting. I could see how quickly they had done the analysis and of the especially the informatics tools they used for deciphering the COVID that two virus and so on. And then I also would like to congratulate Obul Reddy for his special reference to the genomics and regarding the SARS-CoV-2. And Rahul Narang's talk was also very interesting with regard to all the zoonotic diseases, and he gave a very detailed talk on the economic impact of all these. And then especially the last one by Prashant was wonderful. The work they did in the recent times and comparing the COVID CoV1 and CoV2, and especially the open reading frame and differences therein, and to show the differences across the various types of the COVID strains we have and so on. And I really enjoyed thoroughly all the talks. And I would like to say to Prashant that regarding the curcumin work, it's very interesting. And I know Dr. Kavi Kishore is also involved in this work. And I do find that the curcumin would have a great effect in finding some cure, and maybe it will also help in the drug which we administer for COVID. And with regard to the bioavailability which you mentioned, if you use curcumin in the form of a glycoside, it is much more bioavailable than the curcumin itself. So because it can gain entry and then can be easily activated, and the curcumin can reach the targets. So with these few observations, I thank you all for arranging this webinar, and all the speakers and all the participants were excellent with good questions and good discussion. I enjoyed, and we hope we'll continue this with similar or newer topics in future. Thank you very much.
Thank you very much, sir. On behalf of Biotechnology, Vignan University, Professor Ravishankar, sir, for giving valuable information on webinar. Now, actually, I'm going to hand over this further next conclusion section to Dr. N. Jalja, our organizing secretary and our colleague.
First of all, I'm Jaleja. I'm working as Associate Professor at Department of Biotechnology, Vignan University. Thank you, sir. Especially our speakers, Dr. Rahul Narang, sir. It is a very impressive speech, and it is a very informative speech, sir. After that, Dr. Prashant, sir, you also have given a very good information about system biology and in depth. So thank you so much. Dr. Obul Reddy, sir, thank you so much for accepting our invitation. And our head of the department, Professor Krupanidhi, sir, thank you so much. So, and also I would like to thank you all the management of VFSTR and our Vice Chancellor and HOD for accepting our webinar. When we have asked them, they immediately told you can organize. So I'm thanking to Vice Chancellor and HOD. Now I sincerely my gratitude and thanks to my guide, Professor P.B. Kavikishore, sir, now scientific advisor to Vignan University. With sir, we want to grow up very nicely in the research field. Thank you, sir, Kavikishore, sir. So I like to thank Krishna Kishore, sir, for providing the Zoom link, and he is with our technical support continuously in this webinar. Thank you so much, sir, Krishna Kishore, sir. And I like to thank our Dean R&D, Srinivas Rao, and Vijay Ramu, Dean Academics, sir, Dr. Vijay Ramu. And at last, I thank our Rangarao, sir, for coordinating as a convener for this whole webinar from morning until now continuously. Thank you so much, Rangarav sir. So I thank especially all the participants for very actively giving so many messages and continuously listening, and they're posing some questions in the chat box. Thank you so much. If some inconvenience is there, we are really bit sorry for that at the starting. Thank you for the Department of Biotechnology, all the faculty members. Thank you, sir. Now we will close the session.
And those who are participants, we will actually send you the certificates through registered emails in a week time. So please, if you have any questions, please write to me or our organizing secretary. We will get back to you soon. Again, thank you very much for all speakers and also participants. Thank you all. So we are going to close this section.